This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 175, recorded on April 26, 2018. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michele Swanson.
Hello.
How's everything?
Oh, everything is peachy. It's graduation weekend here.
Wow.
So the streets are full of vans being loaded up with things and people dressed up and the occasional cap and gown.
You have nice weather for that?
Yeah, it's. I think right now it's in the 60s. It's sunny.
All right.
Also, it's spring.
Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone. And it's spring here.
70s, 80s?
70s. We haven't gone into full-blown summer yet, so I'm very delighted that it's staying in spring.
Yeah, it's still chilly here, but today it's kind of nice outside. We have nice blue sky. Yeah, the plants are blooming. All the daffodils are out here.
Hooray.
That means it's spring. Now, today being the 26th of February, that is Elio's birthday.
It's April.
26th of April.
Where am I getting February from? Is that what I want it to
be? It's chilly.
26th of April, it's Elio's birthday. Happy birthday, Elio.
Happy birthday.
90.
Woo.
That is amazing.
Who would have guessed?
And he's doing he must be the most advanced in age person doing a podcast in the world. Now, someone will write in and tell us. Most experienced. We're grateful that he's joining us. Anyway, he's away today. He asked for the day off to celebrate his 90th. So
he's having a wonderful family celebration. So
that's great, and he will be back next time. But boy, that is some milestone.
And his collaborators on his blog site, Small Things Considered, wrote a tremendous tribute to him. Very with tongue planted firmly in cheek. And it's really quite spectacular on the Small Things Considered blog. If you haven't checked it out, you really need to see what it says. And the title of that particular blog post is A Whiff of Taxonomy, Eliobacter woozii, or woozii.
That's great.
And it's really quite spectacular, describing some of his phenotypic traits.
So should we link to that?
We will.
Is it appropriate for a birthday?
Sure.
It is
totally appropriate. We will put that in. I'll paste it in right now to remind me. That's good. Thanks for bringing that up. We have a follow-up that's related to Elio. It's from Thao and the TWiM fans on the BBM organizing committee. BBM is the Boston Bacterial Meeting. And they write, we wanted to write to sincerely thank Elio for his endorsement of the Boston Bacterial Meeting during last week's episode. We enjoyed having him as a guest 2 years ago, and we're pleasantly surprised to hear him mention us. Would you be willing to provide a few additional details about our conference to your listeners? Sure. Boston Bacterial Meeting invites you to join a quorum of fellow bacterial researchers in New England and beyond. Visit bostonbacterial.org to register. It's a 2-day conference from May 31st to June 1st. It's organized by grad students and postdocs. Attendance is now at about 550 researchers from hundreds of labs. There are opportunities to attend oral and poster sessions, as well as breakout discussion sessions. And the keynote speaker this year is Diane Newman, professor of biology and geobiology at Caltech. There you go.
Wow.
Awesome.
Can I add, I did both my doctoral and postdoctoral training in Boston, and I had the privilege of giving a talk at that meeting many years ago. And it was right when I was about to go on the job search. So it was really a great opportunity to present my postdoc work to a group and get some feedback. So I highly recommend it.
Yeah, and it's not very expensive. $120 for a 2-day conference. This year it's going to happen at the Science Center at Harvard. So they do have that website, bostonbacterial.org. So if you're in the area, check it out. Thank you, Thao and the BBM organizing committee. And we also have some sad news for you. Allan Campbell has passed away.
Yeah, it's really. I had the opportunity to meet him at the general meeting of the American Society for Microbiology a number of years ago when it was in Los Angeles, and Jeff Miller, who was I guess he was chair of the meetings committee at the time, was gracious enough to introduce me to Dr. Campbell. And you know anything about phage biology or have ever read any of the papers on Lambda, you really are in awe when you encounter the author of the Campbell model of DNA integration into the bacterial chromosome. I mean, it's what we do. It explains everything. And for many years, it was the great frontier.
He was the one who proposed that idea that lambda DNA circularizes and then integrates into the E. coli chromosome. And that's something everybody learns. And he proposed it in 1962.
And it was in a paper in the Advances of Genetics, and it was simply titled Episomes. And he was the sole author.
Mm-hmm.
It was much smaller science back then.
But so influential because we read it now and we just assume it was handed down on a stone tablet, right? But actually there was
absolutely
a group of scientists who had to deduce this mechanism from the elegant genetics that they did back then.
Yeah, so we have a little piece on his work in our textbook, Principles of Virology, which was written by Ann Skalka, who worked with him.
Neat.
And she writes, although the model seems obvious today, it was not so in the 60s. It was an alternative in which linear DNA integrated, but that was shown not to be true. And she writes, lambda DNA integration remains an important paradigm for understanding the molecular mechanisms of DNA recombination and the parameters that influence the joining of viral and host DNAs.
Neat.
He was a professor at Stanford since 1968. Wow.
But he got his start. He was a graduate student at the University of Illinois in Urbana-Champaign,
mm-hmm.
and trained with Saul Spiegelman, but also took a genetics course from Salvador Luria there.
Mm-hmm.
And I was fortunate to be a seminar speaker at Illinois just last week, and they are a proud host of a Milestones in Microbiology designation. So they've been recognized for their long-standing contributions to the field of microbiology.
And in 2004, the American Society for Microbiology honored Dr. Campbell by awarding him the Abbott ASM Lifetime Achievement Award. And in honoring Dr. Campbell, in his award citation, they cite his exceptional insights and achievements in the field of molecular genetics. I guess he's one of the founding fathers of the field of molecular genetics, along with Luria and Delbruck. And a career of groundbreaking research, it says, that has had a profound influence on several fields, including molecular cloning and gene therapy. Dr. Campbell's research has concentrated on the genetics of bacteria and their viruses, especially the integration of viral DNA into host chromosomes. And as Vincent's already alluded earlier, his most prominent discovery was the proposal of the Campbell model of virus insertion, where viral DNA is inserted into the host chromosome, becoming covalently bonded to the bacterial DNA, and then remains dormant until activation. So it's just an incredible body of work that he's left for us all. And there's still much to be learned about phage lambda. It's not completely put to bed.
What I found really fascinating is as I was doing some reading about his life, I came across a piece he wrote in 2012 in the journal Bacteriophage called Life in Science. And he tells the story that after graduating from Berkeley with a major in chemistry, he really wanted to be a novelist. He grew up in a house where his parents had studied English and journalism, but they pushed him towards science. But he wrote that I had no ambition to do research. One of the only reasons, or he said his main motivation for applying to graduate school was mainly a desire to postpone getting a real job. And then he talked about his thesis project and said that he really admired his advisor, Saul Spiegelman, because he thought he kind of tackled really difficult questions. And he wrote that his thesis advisor's efforts from that period ended up having virtually no impact, and neither did my thesis research. I think it's a lesson to all of us that don't think you're behind if you haven't already published, some major paper. People move through science in their careers at different stages, and here he is obviously just a great in our field, and yet he did not have a direct path to that. He was here at Michigan for 4 years, by the way.
Really?
Yeah.
That I did not know.
No, I didn't either, but it's in this little bio. He was here as a lecturer in the Department of Bacteriology. So,
as of this recording, there's not much on the internet about his passing. It has appeared on Facebook. I confirmed it with Ann Skalka yesterday, but there's no obit yet, so I assume that'll be coming in the next few weeks. I also misspoke. Ann Skalka did not work with Allan Campbell. She worked with Al Hershey.
Oh, okay.
It was the same era, more or less, I think. Phage group. All right.
Oh, and one other thing. If you're interested in phage lambda, the organism or virus that he worked with, there's microbiology and molecular biology reviews that's in the public domain called Little Lambda, Who Made Thee by Max Gottesman and Robert Weisberg. And it really highlights how lambda, it's really a nice treatise on lambda biology that highlights some of Dr. Campbell's work in it, and actually the contributions that he's made deciphering it. It gives a little historical perspective of how people did the science in order to work out the intricacies of how this prophage actually does its thing in E. coli. So I'll put that in the show notes for you.
Yeah, Michael, I also appreciated you pointing out this piece that he wrote for Nature Reviews Genetics in 2003, where he talked not only about the basic science that we've learned thanks to the phage model, but also how phage are now being used as tools to as an alternative to antimicrobial therapy, for example, and in phage display, so techniques we can use in the research lab and other things. So it's a really great essay published in 2003, Nature Reviews Genetics. I'll link to that as well.
And Stan Malloy posted on his Facebook account. Stanley is, of course, very much a gadfly of all things cool in science. And he posted yesterday that the FDA is looking to clear phage therapy for the treatment of Crohn's disease. And this is, of course, out of the group out of Georgia, specifically out of the group that was originally founded by de Harel at the Eliava Phage Institute. So it's really, we're seeing this renaissance, and it's a little bit different than our traditional snippet, but I think when one of the founding fathers leaves us, we should pay special tribute to the contributions that they made to our particular field.
Mark Martin says, Allan was on my PhD committee. Mark Martin from Puget Sound was the smartest person I ever knew fairly well. He was so patient with my own 25-year-old self. We weren't friends, but I miss him as a person as well as a one-of-a-kind scientist. Cool. 88 years old.
Yep.
I want to tell you about the upcoming meeting called ASM Microbe, the annual meeting of the American Society for Microbiology. This year it's in Atlanta, Georgia. And ASM has a special opportunity for our podcast listeners. Get $50 off registration for Microbe 2018, which is June 7th through 11th in Atlanta, using the promo code ASMPOD. ASM Microbe 2018 connects scientists with their science and showcases the best microbial scientists in the world. Delve into your scientific niche in 8 different tracks. Don't miss this opportunity. Visit asm.org/microbe. That's asm.org/microbe and use the promo code ASMPOD. It's all one word for $50 off registration. Okay, we have a paper for you today, which comes from Nature Microbiology. The title is Topical Application of Aminoglycoside Antibiotics Enhances Host Resistance to Viral Infections in a Microbiota-Independent Manner. And this comes from Yale University and the Wakayama Medical University in Japan. The first author is Smita Gopinath, and the last author, the PI, is Akiko Iwasaki. Aminoglycoside antibiotics. Now, I think what they were doing here in this paper, they have a model of herpes simplex virus type 2 infection of mice. And this is a genital herpes simplex virus. And they have a model where they inoculate mice intravaginally, and they can watch the virus replicate and spread to dorsal root ganglia and replicate there and spread to other sites and develop disease. And I think they were trying to figure out the role of the microbiome in this infection. They were treating mice intravaginally with a cocktail of antibiotics, including ampicillin, neomycin, and vancomycin. And they found an inhibition of viral replication, because they don't put it that way, but they say to investigate the effect of local antibiotic treatment on genital herpes infection, we treated mice with this cocktail. And to their surprise, the antibiotic
and to my surprise,
mice had decreased vaginal viral titers. Now, I have to tell you a little bit about neomycin because it was discovered by Selman Waxman. And last Friday, I was the 65th Selman Waxman lecturer at a Theobald Smith conference at Seton Hall University. How about
that's
that?
Great. Congratulations.
For coincidence.
Yeah.
So he discovered it, his laboratory discovered it in 1949. And I know neomycin because I have a tube of it sitting right here on my desk, because when you get a cut, you put it on it, and it really prevents infection. It's mostly a topical aminoglycoside that binds the 30S ribosomal subunit of bacteria and inhibits protein synthesis. So you would not think that this would have any antiviral activity, but it does, as you'll see in this paper, it's indirect. It's indirect antiviral. So they have this treatment of mice with this cocktail. They find it reduces viral titers, herpes simplex type 2 vaginal viral titers. So they pre-treat the mice. They pre-treat them for about a week, and then they infect them. That's the protocol. And then it also reduces disease, and antibiotic-treated mice have much lower titers and very little disease pathology. So then they said, which of these 3 antibiotics does this? And they narrowed it down to neomycin by doing infections with single antibiotics. You know, the first question in my virology course is a question designed to trick students. I have to find.
No.
And now they're going to be able to litigate this, Vincent, with this podcast.
I know they're going to say, but you said this. Let me tell you what the question is, because it's, and I say, which of the following statement is true? Number 1, all viruses make us sick and can be lethal. So that's obviously not true. Number 2, our immune system can manage most viral infections. That's not true. 3, humans are usually infected with 1 virus at a time. That's not true. 4, the press is usually correct. Oh, it's not even in this one. It's a different. Anyway, I asked him a question about antibiotics are used to treat virus infections. And of course, I want them to realize that an antibiotic is not used to treat a virus infection. Of course, they could now find this podcast and say, but you're wrong. Well, we'll see. We're not going to treat virus infections with this.
Well, and the authors even basically end their paper by telling us, hey, we don't want you to use aminoglycosides to treat viral infections.
Right. Yeah, because
Speaking
you're
of
going
the
to.
authors.
Yes.
Did you have a chance to say who published this paper?
I did.
Oh, okay.
I guess you were running away somewhere.
I must have been distracted momentarily.
It's okay. Yeah, I mentioned the first and last authors and the institutions. What's going on here? The rest of the paper is trying to figure out what is the mechanism. So first of all, they say, does it have anything to do with the microbiota? Does wiping it out with neomycin or lowering it have the effect. So they take germ-free mice and they treat them with neomycin, and then they infect them with herpes simplex virus type 2. And those mice also have very little virus replication and no disease pathology.
I'll bet that was a surprising day.
Yeah.
And they also do the flip experiment too, where they infect and then they do the flip experiment where they actually show the drug in the germ-free animals that the virus can actually be treated by the neomycin.
Right, after
So
infection.
infect first.
Infect
After-
first and afterward. Yeah, because the previous experiments, they treat for a week to deplete the microbiota, right?
Yes.
And so here, the microbiota doesn't matter because the germ-free mice don't have them. And then you can add the drug after infection, although it's not as good afterwards.
True.
But it does work. All right, so what's going on here? What they did was they extracted RNA from the vaginas of these mice, either treated or not treated, and asked what genes are differentially transcribed. Maybe we'll get an idea for what's going on. So neomycin is doing something. And they find a lot of genes in the type 1 interferon pathway are enriched. They go up when you treat the mice with neomycin. So the interferon type 1 pathway, interferons bind receptors on cells, and then they turn on hundreds and hundreds of interferon-stimulated genes, or ISGs. So these ISGs are what go up. And they say even in germ-free mice treated with neomycin, the ISGs go up as well. So how about
that? And it was not a subtle signature. I was impressed that 30% of the genes that were upregulated fell into the class of these interferon-induced genes.
Yeah, it's quite clear. Something.
Yeah.
So somehow this neomycin is turning on interferon pathways. And these interferon-stimulated genes, those are the ones with antiviral properties, right? So an example was in the old days, we used to treat hepatitis C infections in humans with interferon because we had nothing else. And of course, that turns on interferon-stimulated genes, which have the antiviral effect. Interferon itself is not antiviral, but it turns on other genes. Just like now we see neomycin is not antiviral, but it turns on genes that are antiviral. But you don't want to treat people with interferon because, well,
it's toxic.
It's toxic.
It becomes toxic.
The dosing is tricky.
And you feel very badly. And for Hep C, only a fraction of the people it worked in, and it made everybody feel very badly, so people would stop taking it and so forth. So it's not good. You have to have, and now we
had
have
to give it with antifreeze as well.
Antifreeze.
Yeah, you used polyethylene glycol in concert with
they would pegylate the interferon. Yeah, it's funny the way you said that, Mike.
It's antifreeze.
Yeah, they pegylate the interferon. They chemically conjugate it and makes it last longer. That's right.
Yes.
That's right. But I don't think the way you said it, I thought they were.
Well, no, it's how I remember it's pegylated. It's
pegylated.
How I remember
That's right.
it's
it turns
pegylated.
It prolongs the half-life of the interferon.
in the system.
Right. So here the neomycin is upregulating ISG expression. Now, what's interesting, they did a couple of other experiments. This ISG induction by neomycin is maintained up to 3 days after treatment and is only partially lost after a week. So it's really sustained.
I was surprised by that.
Yeah.
And you don't need much.
You don't. But it is restricted to the site of application because they did not see ISG upregulation in the lungs.
So this was a topical application.
Now, Mike,
that's good news.
Tell me, either of you, I was reading a little bit of neomycin. It's not well absorbed, right?
No, and it never was made to be well absorbed because of all its associated bad side effects it has in people. So it's not all that well absorbed. But there are other aminoglycosides out there that are in clinical use that are well absorbed, but they also have all the bad side effects of aminoglycosides.
Yeah. So then, as Michael said, there are other aminoglycosides. So they looked at a panel of different ones, and they do the same experiment. They ask if they apply it to the vaginal mucosa of these mice, do they induce ISGs? And 5 of the 7 do induce or increase ISG expression. The 2 that do not are streptomycin, which is another Waxman product, right?
Absolutely.
By the way,
won the Nobel Prize for it.
There was a guy at this meeting in Seton Hall last week who was trying to get the New Jersey legislature to name the state microbe Streptomyces.
How many states have state microbes?
2. Wisconsin,
really?
Lactococcus,
Yes.
just for cheese, right? And
cheese.
Oregon has Saccharomyces for beer.
For beer. Wow.
And so, if Michigan had one, what would they have? There's no microbe.
Wait, I'm going to need to give that some thought.
How about South Carolina?
Let me get back to you on that.
You must make something.
South Carolina would probably have a mycobacterium because there's lots of mycobacteria wandering our pine trees.
Right. So the aminoglycosides, getting back to this, 2 of them do not induce, and they have a chemical core that's different, so that may explain that. And then they say that these antibiotics bind ribosomal RNA, and there are other antibiotics that target the ribosome, like tetracycline and chloramphenicol, but they do not induce ISGs when topically applied. So that's very interesting. So binding the ribosome is not all you need to induce ISGs. Now, is this ISG expression actually what causes the antiviral effect? I would say for sure, but they did an experiment to show that. They treated mice with 2 ISG-inducing and 2 ISG non-inducing aminoglycosides. They pre-treat the mice. They infect with herpes simplex type 2, and only the mice treated with ISG-inducing aminoglycosides were protected from infection. So it looks like ISG induction is what is protecting them. They also do a cool series of experiments to ask, would topical application of neomycin protect you at a different mucosal surface? Right, so that's cool. And they use influenza infection of mice to do this, the intranasal infection. And what they do is they pre-treat mice with neomycin, and in fact, it substantially decreases viral replication, increases survival time in mice treated.
I forget, how did they deliver? Oh, intranasal.
Intranasal. Okay. Intranasally.
They probably just topically applied it to the nose with a Q-tip, is my suspicion.
Yeah, I'm looking here.
And I'm scrounging the methods as well.
How about other viruses? They check. So far we have herpes simplex and influenza. They check Zika virus, which replicates in the vagina of mice, and it's known to be controlled by interferon. And they find that the 2 ISG-inducing aminoglycosides do reduce viral replication as well.
And Vincent, these are very different viruses, aren't they?
They are. They're all different. Herpes simplex is a pretty large double-stranded DNA virus, and
it persists. It persists
as well. And influenza is a RNA virus, a negative-stranded RNA virus, and Zika virus is a positive-stranded RNA virus, and they're all inhibited. So that's the power of interferons. They have broadly antiviral activity because you have so many hundreds of ISGs that are induced. So far, all this is done in mice. What about human cells? They take peripheral blood monocytes from donors, which you can just get from someone in your lab if you have the right protocol. And I remember Howard June used to do that all the time. Michele, you must do that too, right?
Oh, well, when I was a technician in Sam Silverstein's lab at the Rockefeller and at Columbia, that's what we did.
And did you have to pay them?
I don't remember paying them.
Because I know Howard used to pay them all the time. He said that was required. You get blood, and then you can purify monocytes, and you can infect. Well, what they did here is they treated these monocytes with these aminoglycosides, and it increases ISG expression in the monocytes. And the control here is poly-IC, which is a synthetic double-stranded RNA, which is known to trigger ISG responses, and that also does that in these cells. They also found that this treatment in monocytes will reduce influenza virus replication. So it works in human cells as well as in mouse cells. Now, what's going on? What is sensing the neomycin? We know that interferon is turned on by any one of a number of sensors like toll-like receptors and cytosolic helicases. And the cool thing is if you're at Yale, you have access to mice that lack the genes for every one of these sensors because that's the home of innate immunity.
Yes.
What was his name?
Leaders.
Richard Flavell.
Yes.
started the field. Yes, they're leaders. And he had this idea years ago, and then he put together a department, including Akiko Iwasaka, that works on innate immunity. So they have mice lacking RIG-I, which is a cytosolic nucleic acid sensor. They have mice lacking DNA sensors, CGAS, and STING. So they could take these mice and treat them with neomycin and ask, do we have an antiviral effect? Do we have ISG induction? And in fact, the mice lacking the DNA sensors, they have no problem responding to neomycin. But if they use mice lacking a protein called MAVs, which is downstream of these cytosolic receptors, then you have reduced ISG production and reduced protection. So it looks like neomycin is going through typical RNA sensor pathways. So that's pretty interesting. So next they say, what is exactly the sensor involved? And they have mice lacking toll-like receptor number 7 and toll-like receptor number 3. These are endosomal RNA sensors. They're proteins on the membrane in the endosome. So when a virus comes in to the endosome and the RNA is exposed, these sensors will sense it and the cell will start producing interferon. They found that TLR7 is not involved in the neomycin effect, but TLR3 is. So somehow neomycin is tickling TLR3 and turning on the interferon stimulated gene pathway. So that's pretty interesting that neomycin would do that. You could not predict that. And that's the way research is, right?
Yeah.
No.
Although we do know that the aminoglycosides bind to ribosomal RNA.
Yep.
So to think about nucleotides was not so far off.
No, I suppose, yeah. All right, but that's not as we found before, though. Binding ribosomes is not enough, interestingly, because the chloramphenicals and et cetera do not have this effect. So
right.
Well, the chloramphenicol hits the 50S.
Yeah.
And remember what the hallmark of these aminoglycosides is that they interact with the ribosome shape at the same time as they're interacting with the mRNA codons. And they're effectively doing two things at once, if you will. And that's the magic of it. And so it's, as you're going to get to the poly-IC stuff and the TLR3 receptor again, it will come into greater clarity as you begin to unfold this wonderful story.
So far, we know that TLR3 is involved, toll-like receptor number 3 is involved in the neomycin effect. They also show that proteins downstream of toll-like receptor 3, including TRIF and IRF3 and IRF7, these are transcription factors that are activated by sensing of nucleic acids. They go in the nucleus and they turn on the synthesis of these ISGs. And they have mice that lack every one of these. It's just great to be in a place where you have all these mice.
Yeah, just to pull the reagent off the shelf
just
and
do it.
experiment.
If you're not there,
this is a lot of work they did. My goodness.
So they showed that. So basically, neomycin is working on this toll-like receptor 3 TRIF IRF3/7 signaling pathway, and that's what turns on the ISGs. So that's the molecular part. Now we get into what Micah would say, the immunology. What cells are responsible for this? And in the vaginal mucosa, you have, of course, epithelial cells, you have resident white blood cells, and you have circulating white blood cells. So they asked whether the induction of ISGs by neomycin requires tissue resident cells or circulating leukocytes. And so they can actually block the recruitment of circulating white blood cells to the vaginal mucosa by pertussis toxin. Basically, pertussis blocks receptor signaling, which prevents chemokine-mediated recruitment to the vaginal mucosa of these cells. That's amazing, right? So you treat treat mice with both neomycin and pertussis toxin, and this ablates the protective effect of neomycin. Yeah.
Well, this also illustrates the insight of having all these knockouts affords these investigators, because if you didn't have all the knockouts at your disposal, you wouldn't have appreciated the subtleties of pertussis toxin and what it's actually going to do, unless you have the opportunity to use the reagent and then be able to ask the binary question.
Yeah. Now, what's interesting here, right? You treat with pertussis toxin and you ablate the effect of neomycin. In other words, there's reduced ISG induction and you get higher viral replication. But what that tells you is that recruitment of white blood cells to the vaginal mucosa is important, and that apparently is done just by topical treatment with neomycin. So I'm surprised by that, because this is not penetrating the mucosa, but I guess just being on the mucosa is enough to recruit these cells, right?
Could it be taken up by endocytosis?
Maybe. It could be, yeah. Maybe that's it. But it's clearly these recruited cells that are doing this. Now, what kind of cells? What specifically are? There would be a number of different kinds of cells that would be recruited by neomycin: monocytes, monocyte-derived dendritic cells, or classical dendritic cells, all of which have cell surface markers that you can use to distinguish them. And they find that the classical dendritic cells are increased on neomycin treatment. So these are being brought into the mucosa by neomycin. So what they do is they isolate these classical dendritic cells from the vaginal tissue of mice, either treated or untreated with neomycin, and they show that the ISGs are increased in those cell types. And so again, these are dendritic cells being recruited to the mucosa by treatment with neomycin. In those DCs, and Michele, those are very good at taking up things at phagocytosis, right? So maybe they are taking up the neomycin, and it's tickling TLR3 in the endosome, right? They treat mice, so they have mice that you can treat with diphtheria toxin and ablate the classical dendritic cells specifically, which is another beautiful mouse reagent to have, right?
They've engineered that.
They have engineered
molecular trick.
So then you give those mice, you give them diphtheria toxin, you deplete the dendritic cells, you then treat with neomycin. Guess what? No ISG expression in the absence of classical dendritic cells. So another piece of the puzzle, classical dendritic cells are essential for this neomycin-mediated antiviral effect. In the classical dendritic cells, there are different subsets, of course. What would immunology be without subsets?
More
cells? Intelligible. It would go on, within each cell type, and they're defined by cell surface markers. And they say they look for within the dendritic cell subsets, ones with toll-like receptor 3 expression, and they find a single subset with high TLR3 expression. These are typically found in the thymus, in the spleen. And they find that this subset is the one that is recruited to the vaginal mucosa by neomycin. It's the one that's blocked with treatment by pertussis toxin. And they can deplete these from mice, specifically this specific subset of dendritic cells. They can deplete those when they treat with neomycin. You don't get induction of ISG. So this is beautiful. They've teased their way through to a specific subset of dendritic cells that come into the vaginal mucosa. Upon treatment with neomycin, they make ISGs and they help clear the infection.
It really is an impressive, rigorous, logical body of work in one paper.
Yeah. What else do we have here? Now, what's the mechanism? That's the last part of this paper here. They say, well, TLR3 is a sensor of double-stranded RNA, and they're thinking maybe these aminoglycosides make host RNA more visible to toll-like receptor 3 in neighboring dendritic cells. So the idea here is, host RNA is not supposed to be recognized by toll-like receptor 3 itself. Toll-like receptor 3 only recognizes foreign RNAs. So what they do is they take splenocytes, which contain these dendritic cells. They treat them with their aminoglycoside. Then they wash them to get rid of the aminoglycosides. And then they incubate them with dendritic cells, the same subset that don't have the aminoglycoside in them, and from wild type and TLR3 knockout mice, and then they measure ISG expression. So you've got neomycin treated dendritic cells. You mix them with untreated dendritic cells from wild type or toll-like receptor 3 knockout mice, and you measure ISGs. And interestingly, if you mix the neomycin, it's not neomycin, but I'm using that one because it's easier to say than kasugamycin.
Mm-hmm.
Kasugamycin treated DCs with untreated DCs, wild type, you get ISG induction in the cells. So they must be taking up something from the other cells. But if you use TLR3 knockouts, you don't get that induction. So they're saying this suggests that the aminoglycosides are binding host RNA and making it better to be recognized by TLR3. And to sort of confirm that, they ask, will synthetic double-stranded RNA augment the activity of these aminoglycosides? And they find that if you add enough double-stranded RNA, you get a synergistic induction of ISGs with the aminoglycoside and the double-stranded RNA. So their idea is that somehow the aminoglycosides binding to the double-stranded RNA and allowing it to be better recognized by toll-like receptor 3.
But there are some topological mysteries here.
Of course.
Where is this double-strand RNA normally?
Yes. So,
well, remember, RNA likes to pair with itself. So it may not necessarily be double-stranded RNA. It may be just message RNA that happens to be pairing with itself.
It could be
forming a partial duplex.
There's also a whole set of RNAs in eukaryotic cells that are transcribed by these highly repeated regions called ALU regions in the genome, and they can form double-stranded RNAs. They are typically modified by an enzyme in the cell called ADAR, adenosine deaminase, and it changes A to I, and so that makes the cell blind to that double-stranded RNA.
Nice.
And maybe the aminoglycosides are reversing that blindness, right? So that's something they could actually explore, which would be interesting. Okay, so that's the story. These aminoglycosides hit a toll-like receptor and turn on ISGs, and they think that it makes cellular RNA more visible to TLR3. Now, they do mention that other antibiotics have been shown to have antiviral activity, and that includes azithromycin, which inhibits Zika virus replication. We don't know the mechanism, but of course, when Zika appeared, everyone tested everything that was FDA approved for antiviral effects. Now, Michael, isn't azithromycin the one that is used to treat typhoid that's left?
Yes, and remember, it concentrates in the phagocytic cell. Its concentration is much, much higher, like 50-fold and higher in the phagocytic cell. So when the phagocyte is effectively cleaning out the environment of, salmonella in the case of typhoid fever, it, of course, will grab onto it. And because the concentration is so much higher, they dispatch the microbe right quick. So, again, this is another mechanism where we're beginning to rediscover antibiotics in this whole new world of eukaryotic biology, where it's beginning to teach us something. These aren't the antibiotics your father used. These are the same antibiotics, but we're now beginning to understand, they're doing something more than just killing bacteria.
So azithromycin and Zika, another one, non-chongmycin. I haven't heard of that one, but blocks entry by inhibiting endocytosis of flaviviruses. And others, macrolides antibiotics have been shown to induce interferons, and there are more. The point is that, as Michael said, these antibiotics can have other activities as well. And what I wondered, which they didn't mention, do either of you think that this ISG expression by neomycin, do you think it plays any role in bacterial clearance by stimulating inflammation, for example?
My bet is anytime you have cytokines being aggravated, the inflammatory response kicks up. It brings, the full force of our adaptive immune system into play, and it can facilitate the clearance. Because one of the great mysteries that you should scratch your heads about is, how do bacteriostatic antibiotics resolve an infection? Because eventually the bacteriostatic antibiotic gets diluted out just simply via, the normal physiological process of clearance. But yet bacteriostatic antibiotics like tetracycline clear infections.
Right.
How quickly? So quickly that it can't be the adaptive immune response?
Well, my suspicion is they're working in synergy with each other. Because
as we see in this paper.
Yeah, as we see in this paper, because you have to dose yourself more than once a day with tetracycline in order to maintain the blood levels, and it has all to do with the area under the curve. But I think this is the brave new area of revisiting our antimicrobials, especially now that we have all this knowledge about immunology that we didn't have when the antibiotics were discovered and licensed.
True, but we also have all this knowledge of the microbiome and how protective it is. And so it's interesting that they do close their paper, as you pointed out, Michael, that they don't advocate the use of these compounds as antivirals. And I don't think we talk specifically about what their toxic side effects are, but they cause some damage in our ears by killing the little sensory cilia, I believe it is.
The hair cells,
Yeah, and is there some other liver toxicity, or is it kidney? I forget which.
Yeah,
kidney toxicity.
Yeah. It's kidney toxicity. That's pretty horrific.
But in thinking about that, they did say that it stays local. So if you administer it just topically, and they found that it works at different tissue sites, different mucosal surfaces, and on different viruses. So, Vincent, what do you think about using Neosporin for cold sores? Do you think that activates this pathway locally?
It's a good question. A cold sore would be on your lip or your nose. So
it's HSV1.
It's a recurrent herpes infection.
It could, but we have a really good drug for that, acyclovir, and that works great. So I don't know that any company would try and get this licensed for a cold sore, right? You'd have to go through clinical trials to do that. And I know that whenever you already have a drug that works really well, it's really hard to get a new one licensed. So I don't know if it would work, but based on this,
Neosporin is already safe. It's on the market.
Yeah. That's an interesting question. I don't know. I would predict it would work because it's herpes virus. It's type 1, but that shouldn't matter. It's interferon induction. Are you worried about, we put Neosporin on our skin all the time for cuts. So we don't worry about the skin microbiome being depleted because it's a local.
Well, there's not very many of them either.
Yeah,
and it'll be replenished.
But so that is a good point. If you don't have, acyclovir, which is I think now it's over the counter probably. You try Neomycin. I don't know.
But acyclovir is oral.
Well, you can put it on you can get topical acyclovir.
Oh, right.
And that's what they probably recommend for cold sores to cause them to go in more quickly.
There are a number of over-the-counter cold sore medications. I wonder if any of them just have Neomycin in them because people have found that it works. Who knows? Well, I don't get cold sores, so I can't try it. So anyone out there wants to try it, but don't blame me if you have a problem.
You're not licensed to practice medicine.
I am not, nor to do clinical trials.
Yes.
So that's interesting. They say we don't advocate the use of this because it will, especially vaginal would affect the commensals, but skin might be okay. However, in a broader sense, inducing interferon stimulated genes we know is toxic with hep C. So I don't think that's not the real message of this paper. I think the really interesting thing is that these aminoglycosides can do something to activate toll-like receptor 3, and figuring that out is really interesting, I think, to find out what the mechanism is. But I don't think we're clearly not going to use Neomycin for treating viral infections unless, as Michele suggests, a cold sore. But
and you have it already in the house.
You already have it in the house, yeah. There are two more things I wanted to mention, which maybe were not clear. First of all, they suggest that dendritic cells, which come into the vaginal mucosa, take up aminoglycosides by endocytosis. Michele mentioned that earlier, but they actually mentioned that in the paper here, and that's how it would tickle toll-like receptor 3, but where it would encounter RNA, we don't know. And they also say the specific RNA is unknown, as you said also, Michele. That's bound.
Yeah. They do suggest that the epithelial cells in the mucosa might phagocytose might take up the aminoglycoside. And then if the DCs then ingest one of those epithelial cells that's loaded with aminoglycoside, then that interaction could happen in the DC endosome.
Yeah. Michele, they did that experiment where they mixed two different DCs, one that had been pretreated and the other not. And they could see ISG expression. But I don't know how they could see which cell was making the ISGs in that experiment, right?
Right.
They have so I'm not sure what that means, except that it's TLR3 dependent. Yeah.
And when they say splenocytes, is that a mixed population of epithelial cells and DCs in the spleen?
Probably a mixed population of lymphocytes and monocytes. I would think. Yeah. Not a lot. I don't think it's a lot of epithelial cells. Mostly lymphocytes and DCs. So,
but the authors do point out that they have more work to do to try to understand where the presentation is happening.
I think that's the interesting part here. How is this mechanistically working? I don't think there's an antiviral here or even an antiviral approach because, as I said many times already, interferon is not really good because it turns on too many genes that are toxic. So the basic science here is what's interesting, I think.
Yeah, interferon is good for the cell that's being attacked. It's not good systemically.
Yeah, that's the problem because it goes systemic and you get all kinds of side effects, right? Including fever and so forth. Anyway, that's that. That's a cool paper.
It's very cool. Very surprising
stuff. Yeah, it is surprising and always good to be surprised.
And very thorough.
It's very thorough. Yeah, it's great. All right, let's do 2 emails. We have 2. One is from Dallas. In your discussions about the chemosynthetic bacteria, you mentioned metabolism of hydrogen, carbon monoxide, and methane. A couple of TWiMs ago. In terms of energy available, methane has both the highest concentration, up around 1.7 ppmv versus 0.5. I don't know if that V is right there. ppm, right? Versus 0.5 for hydrogen and 0.1 for carbon monoxide. Note that these are molar amounts in air, and the weight concentration ratios are even high in favor of methane along with the energy ratios. CH4 to hydrogen is about 3.5 on energy. The HC ratio for methane as the food supply is higher than normal life forms with far more C and O in their makeup, and that must come from carbon dioxide providing a carbon supply. And it gives a link for that. He gives a link to an article, which is called Soil Microorganisms as Controllers of Atmospheric Trace Gases, which was published in 1996. And this was the paper we covered in Antarctica, where they find that these trace gases are made by the soil microorganisms, which are very sparse because it's a very harsh environment. Dallas continues, I didn't bother reading the article, so it may have stressed methane plus oxygen metabolism, but I do know that methane would be the dominant food supply. Methane is highly soluble in membrane lipids and is chemically concentrated, allowing even more efficient metabolism. That is why I consider all the environmental activists' worry about increased methane emissions from fracking pure activist nonsense. To get the methane concentration high enough to be a significant greenhouse gas, the bacteria in soils and water that run on methane plus oxygen as an energy supply would dramatically increase the bacterial concentration and eliminate the methane. If they left out methane metabolism, perhaps they were just being PC, as they know the concentrations and energies. Science shouldn't be PC. It should be what is true, even if it says the methane emissions may be self-limiting with increased emissions just being eaten up by the increased bacterial biomass. By the way, a factory producing single-cell bacterial protein is under construction in Louisiana, I think, using methane and oxygen that is going to be used in fish diets, especially targeting substituting fish meal used in farmed salmon diets. Instead of using food to produce fuel with little CO2 reduction and lots of profits for cronies, the mandatory ethanol production system, this process uses fuel, methane, to produce protein for fish feed, providing human food. It is fully approved feed ingredient in Europe, where they actually have permitted offshore aquaculture to exist as a food production method. With our fracking, we have cheap methane to feed the bacteria. Energy and food are fungible. That's very interesting. Did you get any of that, Michael?
Food is fungible.
I did
Food is
get
energy,
that, and I have one of my colleagues here at MUSC, Hal May, is actually working on a process rather than taking methane out of the ground. He's actually taking electricity and feeding electrons to the bacterium that is instead making acetate, which he in turn converts into food for salmon. And it's because given the amount of solar that we have available and given the amount of wind that's available in very windy countries like Chile, as it goes around the end of South America, it is extremely windy. It's a good way of storing energy because if you convert the electrons to acetate, you, of course, have a much better battery than anything that you could possibly make. And since you're converting free electrons that you get from the wind, which is effectively the ultimate solar energy because that's what makes wind, he's looking at it rather than sinking a hole in the ground and pulling out methane from, a couple of billion years ago.
Neat. Oh, that's very cool stuff.
So he has a very good point. And ppmv is parts per million volume, which is a way they measure gases.
That's right.
It's a volume percent.
So he's basically saying you cannot have too much methane because the bacteria will eat it up or metabolize it, right?
Well, energy is energy. It's like leaving gold coins on the subway. They don't stay there long.
Michele, were you going to say something?
I'm just trying to think about the gas being emitted, from cattle and from fracking and the bugs being in the soil and what concentration of methane is available locally to the bacteria. I'm just having a hard time thinking about this. I could see using the bacteria that can chew the methane to create kind of biofilters coming out of manure piles and fracking operations.
Right. So you're saying that atmospheric methane wouldn't be available to soil bacteria.
That's what I'm trying to. That's what's puzzling me.
Yeah, because there's no way it would be circulating, right? Whatever is close to the ground might be utilized, but higher up in the atmosphere, how would that get down there? I don't know.
But he makes mention of the fact that the methane is effectively can go into the membranes of the microbe, and recovered in that particular way. And so it depends on where the bacteria actually are that is going to harvest the methane. But
I guess the question is how elevated is methane in the soil near a fracking operation?
Right.
Is it, or is it in the air mostly and dissipating? I don't know.
Yep, we don't know.
Well, when DOE did the study of where greenhouse gases were, and they put the satellite over South Carolina, because we have so many pine trees in South Carolina, we have a tremendous amount of volatile organic carbon compounds that are being emitted into the atmosphere from the pine trees. And so it does go up. And you can actually detect it via satellite. So there are concentrations in it. Something like a tree is emitting a greenhouse gas in the terpenes because pine trees make turpentine after all.
Yep. All right. We have one from Greg who writes, I just discovered this on the webpage for TWiM 133, which was an episode where Michael talked about a novel antibiotic from a commensal that grows in the human nose. Long time ago. Fantastic show. Thank you so much for contributing to the enlightenment of the world community. I was trying to retell the story of this novel MRSA defense strategy using lugdunin, which is essentially good bug versus bad bug, and forgot the details told by Michael. So I'll have to listen again, which is not a bad thing, as I often miss things when my mind wanders on the ramifications of a particular mechanism you bring to light and I don't hit pause. My comment is, I think it would increase the audience and usability of the material you present if it were also available as a transcript. Obviously, this is a cost, and it is preferable to have more content than less, but with notes available, perhaps your sponsors could pick up the tab and embed advertising in the transcript, as it would surely increase your audience and exposure. So grateful for all I have learned and the insights you offer. And this is interesting because I got an email also the other day from someone who said, I am deaf and I can't listen, but if you had a transcript, I could read it. So that's very interesting.
I think that technology is coming.
It is coming.
Right. Yeah,
and
that's what I was hoping.
It's just not ready for prime time, because we have been investigating it here at the university, because we put up a lot of content on the web.
Mm-hmm.
And a part of the American Disabilities Act is that we make our content available to the hearing impaired. And of course, transcripts are the best way of accomplishing that. So we've been investigating automatic transcription services, if you will.
Mm-hmm.
The technology is out there, but you still need someone to review it.
Yeah.
And it struggles with scientific and medical words. It's good for conversations, but it really struggles with accents and technical terms.
Also, if you have 2 or 3 people speaking, it cannot distinguish between you, me, and Michele. So you have to go through and annotate it, and that takes time as well.
Yeah, but point well taken.
Yeah, I think it's a great idea. And I have looked into manual transcription services where you pay someone. It's very expensive. It's more than we can afford. So if we could pump the audio through a program that did most of it, and probably in a couple of years it'll be ready, right, Michael?
Well, I mean, look at your cell phone now. It's effectively transcribing your voicemails into text and sending you a text message. So it's getting there.
Yeah.
It's not perfect, but
and mine, by the way, my voicemail coming to my landline here in my office, it's a mess. The messages make no sense whatsoever.
Yeah. Can you imagine an episode of TWiM, right?
Yeah.
All right. TWiM is on any smart device that you have that you listen to podcasts, your phone or your tablet. You have an app that plays them. You can just subscribe. So please do that so you get every episode. If you like what we do, consider supporting us financially. You can give as little as $1 a month. It would really help us. You can go to microbe.tv/contribute for how you could do that, and send us your questions and comments. TWiM@microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you. It was a pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you.
I'm Vincent Racaniello. You can find me at virology.ws. I would like to thank ASM for their support of TWiM, and Ray Ortega for his technical help, and Ronald Jenkins for his music. You can find that at ronaldjenkins.com. Thanks for listening, everyone. See you next time on This Week in Microbiology.